Good morning, everyone. Myself, Mrunal Samir Date, is going to explain you today about the gastric proton pump, also called as the hydrogen potassium pump or hydrogen potassium ATPase. In one sentence, I will define it as transport of hydrogen ions in stomach lumen. This is also seen in lysosome, so let's take a look of it. Gastric proton pump structure determined. The highly acidic environment in the stomach is essential for the digestion. It acts as an important barrier to the pathogens invading via the oral route. However, excessive stomach acidification leads to ulcers. Although this is not a life-threatening condition, it can considerably impair the health of affected individuals. Acid separation in combination with antibiotics is the recognized treatment to eradicate the bacteria Helicobacter pylori, a risk factor for gastric cancer. This environment of pH 1 is regulated by the gastric hydrogen ion, potassium ion ATPase, a class of enzyme that catalyze hydrogen ion transport from neutral cytosolic solution of pH 7 to the acidic gastric lumen of pH 1, fueled by the cellular energy source ATP. Hence, gastric hydrogen ion, potassium ATPs are prominent targets for drugs that treat excess stomach acid secretion. In the figure 1, we can see the gastric lumen with pH 1. This is none other than the gastric proton pump, also known as hydrogen potassium ATPase, is expressed on the surface of the stomach to secrete the gastric acid indispensable for digestion of proteins and food. However, too much acid secretion induces ulcers, which can cause a problem. The gastric proton pump uptakes protons from the neural cytoplasm, which has a pH of 7 to the acidic milieu of the stomach with pH 1, driven by the energy source generated by the mitochondria, ATP. Thus, it is an important drug target for acid-related diseases. In the figure 2, we can have a look at the structure of the gastric proton pump, which is also called as hydrogen potassium ATPase. The alpha subunit is shown in pink, whereas the beta subunit is shown in blue. Biological function and location of hydrogen potassium ATP. The gastric hydrogen potassium ATPase, commonly called as hydrogen potassium ATPase, is the proton pump of the stomach. It exchanges potassium from the intestinal lumen with the cytoplasmic hydronium and is the enzyme primarily responsible for the acidification of the stomach contains and the activation of the digestive enzyme pepsin, which is responsible for the digestion of proteins. This ATPase is found in the parietal cells, which are highly specialized epithelial cells located in the inner cell lining of the stomach, called the gastric mucosa. Parietal cells possesses an extensive secretory membrane system, and the hydrogen potassium ATPase is the major protein constituent of these membranes. A small amount of hydrogen potassium ATPase is also found in the renal medulla. Mechanism of the gastric proton pump. The key player in acid secretion is the hydrogen potassium ATPase or the proton pump located in the cannular membrane. This ATPase is magnesium dependent and not inhibitable by OAB. The current model for explaining acid secretion is as follows. The hydrogen ions are generated within the parietal cell from dissociation of water. The hydroxyl ions formed in this process rapidly combine with the carbon dioxide to form bicarbonate ion. This whole reaction is catalyzed by carbonic anhydrase. The bicarbonate is transported out of the basolateral membrane in exchange of chloride. The outflow of bicarbonate into blood results in a slight elevation of blood pH, known as the alkaline tide. This process serves to maintain intracellular pH in the parietal cell. The chloride and potassium ions are transported into the lumen of the cannulicus by conductance channels and is necessary for the secretion of the acid. Hydrogen ion is pumped out of the cell into the lumen in exchange for the potassium through the action of proton pump. Potassium is thus effectively recycled. Accumulation of osmotically active hydrogen in the cannulicus generates an osmotic gradient across the membrane that results in outward diffusion of water. The resulting gastric juice is 155 and potassium KCl, that is 15 mm with a small amount of NaCl. The figure is of the potassium binding site outside the lumen where the potassium is recycled by the stomach. Genes and protein structure of hydrogen potassium ATPase. The hydrogen potassium ATPase is a heterodimeric protein, the product of two genes. The gene ATP4A encodes the alpha subunit and is about 1000 amino acid protein that contains the catalytic sites of the enzyme and forms the pore through the cell membrane that allows the transport of ions. Hydronium ions bind to two active sites present in the alpha subunit. The alpha subunit also has a phosphorylation site. The beta subunit of this pump stabilizes the alpha subunit and is required for the function of the enzyme. The beta subunit prevents the pump from running in reverse, and it also appears to contain signals that direct the heterodimer to membrane destinations within the cell. Although some of these signals are subordinate to signals found in hydrogen potassium ATPase alpha subunit, the structure of hydrogen potassium ATPase has been determined for humans, hogs, rats, and rabbits and is 98% homologous across all species. Moving on from the gastric proton pump to the lysosomal proton pump. Today, it is universally recognized that lysosomes, the intracellular digestive organelles, show an internal pH approximately 2 units below that of their environment. This not only provides favorable conditions for the activity of lysosomal enzymes which have acidic pH optima, but also assists directly in the digestive process by denaturating ingested proteins to make them more susceptible to enzymatic proteolysis. In other organelles, internal acidification serves a variety of more subtle purposes: the dissociation of ligands from their receptors in endosomes, the accumulation of catecholamine in storage granules, and the processing and sorting of newly synthesized proteins in the Golgi are processes that are regulated in part by intra-organelle acidity. The transmembrane pH in these organelles is generated and maintained by an ATP-dependent proton pump residing in their organelle membrane. The above figure is of a vacuolar type ATPase, the VATPase, initially identified in yeast and plant vacuoles, pumps protons into the lumen of organelles coupled with ATP hydrolysis. This is the main component responsible for the hydrogen potassium pump inside the lysosome. The works which I collected for this PowerPoint presentation are from NCBI, Springer, Wikipedia, and Technology Networks. Thank you.